Thank you for the opportunity. I'm Shailee Jain. I'm a postdoc in Dr. Eddie Chang's lab, and today I'm going to be telling you about our work on using deep neural networks to understand the role of single neurons in the human brain during speech processing. One remarkable ability of the human brain is its ability to be able to perceive and produce speech almost effortlessly. Over the past decades, neuroscientists have been very interested in understanding how the brain is achieving this, and we've really thought about it by thinking about the types of information that are represented in the speech signal. So at the lowest level, you might imagine that the speech signal has a lot of really fast spectrotemporal modulations. At a slightly higher level, there might be a brain region that cares about the individual sounds or the phonetic information in speech, so sounds like f or m or b. At a slightly higher level, there are phonetic sequences that give us syllabic structure. And finally, there are individual words that the brain needs to recognize in any speech utterance. Now, this is a really nice and clean temporal hierarchy, and for a long time, speech neuroscientists believed that this hierarchy maps on very, very cleanly to different brain regions, each of which process information at one level and then pass it on to the next. As nice as this sounds, a lot of studies over the past decade have actually told us that the picture is much more complex than this. What we've actually found is that one critical brain region, known as the superior temporal gyrus, represents information at all of these levels. Different local neural populations in the STG are capable of representing or responding to different types of speech information, be it phonetic content, syllabic boundaries, lexical structure, or spectrotemporal modulations. And more importantly, all of these features are represented simultaneously and continuously. Although this is a really remarkable paradigm shift in how we should think about speech in the brain, all of the evidence we have about the STG currently comes from studies that use tools like ECOG or fMRI or MEG, and can only really tell us about the role of tens of thousands of neurons aggregated across the population. But to understand how the STG might be representing information at so many different temporal scales, I think we need to look under the hood. And that's why our lab has been using neuropixels probes in humans to understand the role of single neurons and local neural circuits during speech processing. So these neuropixels probes are inserted in neurosurgical patients who are awake during brain surgery, and we present them really, really simple English sentences that they listen to. The data I'm showing you here comes from an earlier study that includes 7 different sites across 6 different patients in the STG. And a lot of these neurons show remarkable tuning for many, many different speech features. Let me show you one example. So here I'm just going to, on the top, you're seeing the speech stimulus that is presented to the participant, and on the bottom, you're seeing the spike rasters of one single neuron. X is time, and Y is the number of trials that are presented. The same stimulus is presented several times.
What on earth's the matter with you? Junior, what on earth's the matter?
So you can see that this neuron has very stable spiking patterns for this particular stimulus. And actually, I picked this example to demonstrate how clean and interpretable some of these neurons are. This neuron, for example, has very clear spiking activity right after nasal sounds. So when sounds like mo or no occur in the speech sentence, it produces a burst of activity. This is just one example. We actually found, or the first study found single neurons that tune to every single speech feature that you can imagine or that has been defined in linguistics. There's a lot of heterogeneity, and it's across the entire cortical depth. But there's a lot of these neurons are actually very peculiar as well, including this one. So you might have noticed that it does respond to nasal sounds, but not always. The n sound in the word junior does not elicit the same spiking activity. And I was interested in figuring out why that is. One really important clue that prior models of speech processing are missing is the fact that speech is actually not just a discrete process of just collecting beads in a string. It's actually extremely contextual. There are many, many different types of contextual cues that exist in speech, be it acoustical or lexical, and it's highly possible that a neuron, even with such clear tuning, is actually representing nasal sounds in the context of when those sounds occurred. And I think context is really important to consider if we are trying to strive towards a more comprehensive neural speech framework that can account for the fact that speech is a continuous contextual process. It's not just a discrete representation of individual phonemes stuck together. And this is also important to consider if we want to figure out how the STG is representing simultaneous features at many, many different temporal scales. As you might have guessed from my title slide, There's another system, which is not the brain, that is actually capable of learning extremely rich representations of speech by thinking about it in terms of a continuous contextual process. And that's a deep neural network. I'm just showing you the architecture of one network here. This is UBERT, but it doesn't really matter. You can pick your favorite deep network. It could be Whisper or WaveLM. But the idea is that deep neural networks don't operate on just discretized units of speech. So you don't just give them individual phonetic labels. You give them the entire continuous raw audio waveform, and you allow them to discover discretization. You allow them to discover structure in the speech signal. And this really enables us to study continuous contextual processing, so we can use the internal states of this deep neural network to see if the information encoded in those internal states is somehow related to what the neuron might be spiking towards. So to just describe that framework really quickly, what we're basically doing is we're taking the speech stimulus that is presented to the patient. We're putting in snippets of that speech stimulus into the DNN. Here I'm sampling the hidden states at every 10 milliseconds or 100 hertz, but we're not just putting in that 10 millisecond snippet, we're actually also giving it context from the prior sequence, or the prior sequence of frames. So here I'm showing 800 milliseconds of context. Once you've passed that into the speech DNN, you can extract these really rich, nonlinear contextual representations. And then once you have that, all you need to do is learn a simple linear regression from the contextual features to the spiking activity of each neuron individually. So this really gives you a, this gives you a DNN-based model of single STG neurons. Before I actually tell you what is the utility of this framework, let me quickly tell you how well this works at predicting neural activity. So here on the x-axis, you're seeing the test set performance when you use very simple linguistically motivated features. So for example, discrete categorical representations of phonemes or the pitch of a sound. And on the y-axis, you're seeing these more continuous contextual DNN representations. And what you might find is that most of these STG neurons are at least as well, if not better, predicted by the DNN representations. In fact, about 35% of them are significantly better predicted with the DNN than these more discretized representations. But the point here is not to find the best predictive model of single neurons. The point is to explain what these single neurons are doing. So let's move beyond prediction performance and try to think about how the DNNs actually allow us to manipulate context in a way that is not possible with simple discretized linguistic features. So instead of just taking information at a specific amount of context, we can actually vary the amount of context the DNN sees. It could be 20 milliseconds, 50 milliseconds, 1 second, what have you. And then you can see how these different amounts of context modulate the prediction performance of every single neuron. So on the x-axis, so this is a histogram, of course. That's the y-axis is the number of neurons. And the x-axis shows you the improvement in prediction performance on a test set when you use features that have at least 200 milliseconds of context or beyond. And I use 200 milliseconds because that's almost the average duration of words in our data set. So you're trying to see whether adding context about at least the prior word, if not longer, helps in predicting a neuron spiking activity much better. And what you see is that there's a really broad distribution of context sensitivity in STG. So there are some neurons that are very, very sensitive to context. They actually benefit from extremely long amounts of context. But there are neurons that couldn't care less. And I can actually break this history. I should say that about 40% of these are significant. So 40% of them show significant differences, but there are 60% that are not benefited by context. And that's already interesting in terms of the diversity you see in STG. So our first finding is that STG neurons show varying degrees of context sensitivity. But I can break this graph down to look at how context interacts with the types of features that each STG neuron might represent. So what I'm showing you here is two different histograms, exactly like the one before, but the one on my right only comprises neurons that are tuned to phonetic content, like the nasal neuron I was showing you before, and the darker pink histogram shows you neurons that tune to pitch information. So they could, for example, have a burst of activity when there's a high pitch or a low pitch or some change in pitch that they really like. And what's important to note is the diversity in context sensitivity, even for neurons that have the same feature tuning. So there are some neurons like this that are either tuned to phonetic content or to pitch, but don't really care about context. And a hypothesis could be that these neurons are feature detectors. But there are also some neurons that tune for acoustic phonetic information or pitch, but also exhibit a great deal of improvement in performance with context. And I could hypothesize that perhaps these neurons are doing feature integration. They're actually doing some computation in that context window that's allowing them to represent the phonetic content or the pitch information in a specific way. The last result that so like I said, there is varying degrees of context sensitivity even with similar feature tuning. But given that this diversity exists, the last thing I want to show you is how the context sensitivity might manifest across the population. So we did this really simple decoding analysis where we take the neural spiking patterns across a bunch of neurons at a specific time point, and then we try to see how well you can decode the spectral content of this speech stimulus at different points in the past. And all we want to know is that if there are long context neurons that integrate over long spans of time, are you, for example, able to decode the spectral content way in the past? So are long context neurons actually retaining or creating a memory of their speech stimulus in their representational states? And that is basically what we find. So on the x-axis, you're seeing the different decoder lags. The shortest lag is the minus 100 milliseconds to 300 milliseconds. So whether you can decode 100 milliseconds back in the past, all the way up to trying to decode 700 milliseconds in the past. And you see that the context-insensitive neurons, the blue bar, has a very fast degradation in performance when you try to decode past information. But the context-sensitive neurons are able to decode spectral content even 700 milliseconds in the past, which is consistent with multiple words in our data set. So what this really tells us is that the contextual neural population can track information at very long word-level scales, even in STG. So given everything I want to tell you, I want to put forth our current working hypothesis for how speech really works in the STG using DNNs. Firstly, I think single neurons in STG tune to speech features at very, very different context lengths, and perhaps this helps these neurons integrate information rapidly and give rise to our perception of speech. Secondly, non-primary speech cortex in humans, or STG, is much more sophisticated than a set of feature detectors. So if somebody tells you that they think the STG does just feature detection in a discretized way, don't believe them. And the last thing is the context-sensitive representations within the STG population suggests that there must be a lot of local circuit processing. So it's kind of remarkable that this one specific part of the brain is representing so much about speech at so many different temporal scales. And so we hypothesize that perhaps there's a local circuit or a local recurrent mechanism within STG that is enabling it to represent information at so many scales in a continuous and simultaneous manner. We're yet to test this. This is a working hypothesis, but I do think that our results are pointing in this direction. So yeah, that's it. Thank you so much for listening to me, and I'll take questions.
We have time for a couple of questions.
Hello.
Hi.
Thank you for a really interesting talk. And I have a question kind of about the last point that you raised about the local circuit mechanisms. Mm-hmm. And so, I think many people's experience using neuropixels is really that the signal you get is hyperlocal. And so I guess I was wondering, on a single session, what's the diversity of coding that you're actually seeing? And is it kind of the case that on one insertion, is it you're kind of seeing one coding motif, or is it kind of always intermixed all the time?
Yeah, that's a great question. So in one single insertion, we actually see a lot of diversity. And there are some biases depending on where that insertion is anatomically. But in general, you see extreme amounts of heterogeneity in feature tuning.
Thank you.
Yeah.
Let's have the next speaker come up while we have one last question in the back.
Hi, super cool. I know that there's various behavioral work where the same phonemes will be interpreted differently in different contexts, which seems to match what you were saying about these pitch acoustic neurons also having some context behind them. So I was wondering if you looked explicitly at those behaviorally defined sets, or if that would be something interesting to look at.
Yeah, thank you. I think that makes a lot of sense. I think there's a lot of behavioral and psychometric work on how the context can modulate our perception of different units in speech, but we don't really know the neural mechanisms of that. And for a long time, we postulated that there must be different anatomical regions that would be involved in a process like that. But I think our work is showing that that context is encoded at the level of single neurons in one site of the brain. So we're a little far off from figuring out the behavioral link, but I think it's a really interesting direction to explore.
Let's thank the speaker again. And now we will hear from Jacob Ratliff.
All right. Hi, everybody. I'm super excited to be here. I'm a PhD student at Albert Einstein College of Medicine, and I'll be telling you about my thesis work on long-range inhibitory neurons in the neocortex and how they're important for cortical synchronization and sleep.
Jacob, can I ask you, is that the color you expect?
It's a little... Yeah, it might be. It's probably my flux, actually. There we go. Wrong time zone. There we go. So yeah, excited to be telling you about my work. So there are two major themes that my work focuses on. One is the regulation of behavioral states, which, as you might be able to guess here, the most dramatic example of different behavioral states is the difference between sleep and wake. This has profound impacts on the behavior of an animal, as well as their brain activity. The second theme is the diversity of inhibitory neurons in the neocortex. I'm showing you here an image from Ramonica Hall, who is an early explorer of this diversity, showing the variety of different forms that inhibitory neurons can take in the cortex. We've come a long way since then. In particular, the advent of single-cell RNA sequencing of brain tissue gave us a lot of understanding. When these experiments first started to be done, they confirmed some things that we knew, that we could group different inhibitory neurons based on the genes that they expressed, and we had some idea of what these groups might look like. But there were also some outliers in this data. In particular, this population showed up repeatedly across data sets. This is a really unique group of cells that were defined by the co-expression of somatostatin, SST, and neuronal nitric oxide synthase, NNOS. These are the long-range inhibitory neurons from the title of my talk. They have these really unique interareal projections that are in stark contrast to other canonical locally projecting inhibitory neurons, such as the somatostatin expressing Martinati cell that I'm showing here. And in addition, they've been around for a long time. People have sequenced tissue from humans, from primates, from mice, even from reptiles and amphibians. And across this 350 million years of evolution, these cells have stuck around despite being such a small population that evolution could have gotten rid of. So we wanted to understand what the function of these cells were, and there was some work in the literature to lead us at the beginning of this project. In particular, people believed that these cells to be active during intense periods of slow-wave sleep. The histological marker of activity FOS strongly co-localizes with somatostatin NNOS cells after periods of intense sleep, but not during wake. This, however, is a single point in time. This is one animal that's been sacrificed, and we know that that behavioral state changes dynamically from moment to moment that an animal's around. And one way that you can measure this is by looking at the pupil diameter of an animal. As the animal is more quiet, less aroused, pupil is dilated, and as the animal becomes more alert, pupil dilates. This also changes the brain activity of an animal. During quiet states, such as sleep and quiet wakefulness, cortical activity is dominated by low-frequency synchronized activity, and as it becomes more alert, this activity becomes more desynchronized. So taking this lens, we want to answer a few questions about what these long-range inhibitory neurons are doing. We want to know where they're having their action, what their downstream targets might be. We want to know when they're active, what their patterns of activity are, and we want to know what they're doing in the cortex. So to start out answering these first questions, we need a way to target these cells. We use intersectional genetics, which express a tool at the intersection of Cre and flip expression. We use somatostatin flip and NNOS Cre mice, which allows us to express these tools in our long-range inhibitory neurons. So one of the first things we did is try to figure out where these cells project to. Here is an image where we've labeled these somatostatin nNOS cells with virus injections in both hemispheres. I hope you can appreciate that there's both dense local arborization as well as long-range projections that travel all over the neocortex. We quantified this arbor and found that in the local area around the injection site, this arbor is across all layers. And then as you look long distances away from the injection site, this arbor likes to travel in superficial layers. We also registered this data, the Allen Common Coordinate Framework, to look at which areas these cells project to. And when we label neurons in primary visual cortex, the arbor prefers to go into other visual areas. But it also stretches all throughout the neocortex, except for the most dorsal and the most frontal and most ventral regions, and all within ipsilateral neocortex. There's a small projection to the subiculum, but otherwise the rest of the brain is spared. We also patched some cells downstream and found that these cells target many different neurons. So there's a really broad cell types and areas that are targeted by these neurons. But we also need to know when they're active and when they can influence their downstream partners. And to do this, we turned to calcium imaging. We expressed GCAMP calcium indicator, to measure activity of these long-range inhibitory neurons, while measuring multiple aspects of state, LFP, EMG, facial movement, locomotion, and pupil, to get a really tight handle on the state that the animal was in. When we perform all these measurements simultaneously, it looks something like this. To begin to walk you through this data, when the animal is in a much more low aroused state, when the animal is either sleeping or sitting quietly on the treadmill, these somatostatin nNOS cells are really active. The population is extremely correlated, and in particular, when the animal's asleep, the cells are almost never off. Next, if you look at the periods where the animal is much more aroused, where it's locomoting and actively whisking, these cells are strongly suppressed across the entire population. And then if we look finally at REM sleep, which looks a lot like this, where the cortical activity looks a lot like this aroused state, these cells are also suppressed. So to drive this point home, cells are most active during non-REM sleep, then quiet wakefulness, then strongly suppressed during movement and REM. They also correlate with the activity of these cells correlates with our measures of state, as we expect, where they're positively correlated with measures of low arousal and negatively correlated with measures of high arousal, with delta power, or the synchronization state of the cortex, being the most predictive of their activity. So this answers the question of when they're active. Now we want to know what they're doing in cortical circuits. To do this, we turn to channel rhodopsin manipulations. We express channel rhodopsin in our somatostatin nNOS cells. We recorded from the cortex around the injection site with laminar silicon probes, allowing us to record across all layers, getting local field potential and single unit activity while we stimulated our long-range inhibitory neurons. The first thing we looked at is the changes in local field potential power. We found that we were able to increase delta frequency power, particularly in deep layers, by stimulating these cells. We looked across states, and across all the different states that we recorded, sleep and waking states, these cells increased cortical synchrony. We also looked at our spiking activity. Here I'm showing you the average pairwise cross-correlogram of all the spikes that we recorded from, and optogenetic stimulation caused these spikes to fire more synchronously with one another. We looked at how these spikes fired in relationship to ongoing local field potential fluctuations, and found that spikes are more likely to fire in phase with delta frequency activity. And finally, we looked at downstates, which are a feature of non-REM sleep that are characterized by a positive deflection in the local field potential, as well as a pause in spiking activity. And when we stimulated somatostatin nNOS cells, these downstates became more intense. They became higher amplitude, as well as having longer duration. So taken together, it looks like we have a cell type that when we activate it or when it's active, makes the cortex seem more asleep, more synchronized. But this presents a problem based on some stuff that we knew from the literature. I'm showing you here a figure from a review of the circuits that are proposed to be involved with the regulation of sleep. And for anyone who might not be familiar with mouse brain anatomy, these are all subcortical circuits. There's a really strong history of studying these circuits below the cortex and examining their role in generating sleep. But we have a cell that looks like it makes the cortex more sleepy. So we wanted to test whether or not these cells would generate sleep. So in collaboration with a postdoc in the lab, Geoffrey Terral, we injected across the entire neocortex, 22 different injection sites, a virus that will express an excitatory dread receptor in all of our somatostatin nNOS cells while sparing anything subcortical. We put these animals into their home cage while measuring LFPs and movement to be able to determine the sleep state of the animal. We injected them with either a control vehicle injection or a dread agonist to activate our somatostatin nNOS cells. And as is pretty clear to see, these animals fell asleep much sooner when we injected them with the dread agonist. And across all of our cohort, these animals fell asleep sooner and stayed asleep longer with activation of somatostatin nNOS cells. They had greater periods of non-REM sleep and REM sleep with decreased periods of wakefulness. And this, in concert with a few other pieces of literature that have come out recently, is really redefining the cortex's role in the generation of and maintenance of sleep. With that, I'll summarize what I've told you. During more alert states, somatostatin nNOS cells are largely inactive. Cortical networks are desynchronized, as somatostatin nNOS cells can't act on these local circuits. However, during quiet states, such as quiet wakefulness and non-REM sleep, somatostatin nNOS cells are highly active. The cortex is synchronized, as somatostatin nNOS cells are able to act on these circuits. We can optionetically activate these cells and break this relationship between state and cortical synchrony, increasing synchrony during periods where we might not otherwise see it. And altogether, this paints a picture where these cells are reading out the more broadly generated behavioral state of the animal and influencing local cortical states to match this. But it's not just that there's a readout happening from state to somatostatin nNOS cells. These cells are also feeding back onto the more broadly generated behavioral state by promoting sleep. And with that, I'll thank lab and collaborators who are involved with this and take any questions.
We have time for some questions. One down here.
So I'm just wondering if you know anything about how those hypothalamic nuclei interact with these neurons? Like, are these neurons modulated by histamine or anything?
Yeah, that's a good question. So they express a lot of receptors for hypothalamic peptides. We tried some rabies tracing from these animals to try and see if anything like that popped up. So far, we don't have any great hits.
If we can have the next speaker come up, well, we have one last question from.
What do you think is the pathway through which these cortical cells communicate to the subcortical sleep networks, presumably which are still involved when these cells drive the sleep?
Yeah, it's a good question. So there's some other work out suggesting that long-range projecting somatostatin cells from prefrontal cortex will project to sleep-promoting areas. But it's not really clear whether or not they're the same population. So I think future work will have to sort it out.
Let's thank the speaker again. Now we will hear our last talk of the day by Kermani Nejad.
Okay. Hi, my name is Kevin, and I'd like to first thank the organizer for giving me the opportunity to talk to you about my ongoing PhD work on learning in neocortical microcircuits. which might give some of you a feeling of deja vu. So self-supervised learning has been very successful in machine learning. In image, that's my version of B, by the way. In image, you could just mask part of the image and try to predict the entire image. In video, you can pass the network the previous frames and ask it to predict the incoming frames. In audio, you can pass it the previous snippet of audio and then ask it to predict the incoming audio. And similarly, in language, the next word prediction and next organ prediction has been very successful. For example, brain makes the mind makes the person. But now the question is how the brain can implement such a self-supervised algorithm which allows it to process different types of information. So neocortex is widely believed to be responsible for our cognitive abilities, but its cellular organization and layer structure is very similar across the species and cortical area. So if we zoom in, we can see that the neocortex is made up of 5 to 6 distinct layers that are organized in this cortical column. But understanding the exact computation that can be implemented in the circuit that allows the circuit to process different types of information has been a central focus in neuroscience. For years, a feed-forward canonical microcircuit view has been dominant. In this view, the input from thalamus goes to layer 4, then flows up to layer T3, then down to layer 5 before leaving cortical column and go to other cortical area. But obviously, this is not the full picture of the information flow. Let's look at this study by Constantinople and Bruno that they looked at the postsynaptic potential latencies in different layers of microcircuits. So what they observe is that layer 4 and layer 5 have very similar latencies, which suggests that layer 5 received direct sensory information from thalamus. And just note that there's a delay of activity here in layer T3 compared to layer 4 and layer 5. So this challenges that feed-forward view now that we have a stream of sensory information into layer 4 and layer 5. But this makes the circuit even more complicated. So what could be this algorithm that can basically be implemented in this circuit? We hypothesized that despite its complexity, this circuit is very well suited for temporal self-supervised learning, such that the direct thalamic input to L5 provides this circuit with a teaching signal or a target. But the other pathway from thalamus to layer 4, which is then delayed by the time they get to layer T3, and down to layer 5 makes a prediction about the input. So now we have the prediction of the input, and we have the representation of the input in layer 5. Now we could compare the prediction with the actual input. We modeled this circuit using artificial neural network, and for each of the cortical layer, we used a linear transformation followed by a nonlinearity. We train a network to minimize prediction error, which we also call self-supervised loss, and to prevent the degenerate solution or representational collapse, we use a linear decoder which reconstructs the input from the output of layer 5. To just show you how the circuit can sequence the sequential data, let's look at this example. So the input is now this visual pattern of a stripe with at each timer step it changes orientation based on the top-down input. At timer step 0, the input is in thalamus. At the next timer step, that input is now present in layer 4 and layer 5. At the next timer step, layer T3 receives this delay input from layer 4. It incorporates the top-down information and can make a prediction about the input and send it down to layer 5. So next, we look at how the model can learn a realistic task. To do that, we designed this Gabor orientation task where the input is this images of this pattern with certain orientation. At each timer steps, for example, if the top-down is positive, we rotate the orientation by clockwise by 18 degrees. If it's 0, there will be no rotation. And if it's negative, we make a counterclockwise rotation to input. After we train our network on this task, we wanted to see first, what did the network learn to make the prediction about its input, given the previous input? And second, if it encodes useful information about the input. To do that, we train a linear classifier on the output of layer T3, given the previous input, which I show in the x-axis. So basically, class of every input is defined by the orientation of the Gabor. So let's look at the sample that I showed you in the previous slide. So when the previous input had this specific orientation, layer T3 correctly predict that the orientation of the next input is not going to change. When the top-down was negative, it predicted the next input is going to have an orientation which is rotated counterclockwise. And similarly, when the top-down was positive, it predicted that the orientation of the next input is going to change. And similarly, we fit a logistic regression, the output of layer 5, and we basically summarize the classification accuracy for each class of the input for layer 5, which shows that layer 5 also lends a useful information about the input. But what are the other benefits of having this self-supervised objective in the circuit anyway? Well, one of those is that it helps the circuit to ignore the noise. So to test that, we add some Gaussian noise to the input, and then we train our model, one with the predictive component and one without the predictive component. So as you can see visually, the full model, the reconstruction of the input from full model shows less amount of noise compared to the model in which we knocked out those weights from layer 2, 3 to layer 5, which carry out the basically prediction in our model. We also looked at quantitatively how this encoding of the noise increases in the model by looking at, as we increase the amount of noise, by looking at the reconstruction residual. So as we can see, the full model scales much slower than the model without predictive components. And to get the optimal denoising performance, we trained a denoising autoencoder, which I showed in a dashed line. But another type of noise can be thought as occlusion. Is our model going to be robust to occlusion? So to test this, we modified our previous task slightly, such that now at each time we randomly occlude part of the input. We also move the input randomly by 4 pixels in any direction. After training a model, we see that layer T3 can perfectly decode the class of the input, even better than layer 5, which could be because it's predicting, so it encodes probably better information about the input. But that predictivity completely go away when we apply the self-supervised components of our model. And on the right, I'm showing you some reconstruction from 3 samples. So we can see layer 5 only encodes a visible part of the input, while layer T3 can reconstruct the entire input, but not only that, you can see this blurry heat map shows that it has also uncertainty about where the input's going to be. Well, so far I showed you that self-supervised learning can be implemented in neocortical microcircuits. I showed that you can learn a sequential gap-or task and being robust to noise and occlusion. But it's also important that we show whether experimental data support a hypothesis. So one of the main components of our model, as I said, is this weight in these synapses from layer 2-3 to layer 5 that basically carries a prediction from the superficial layer to layer 5. We can show that the synaptic, the plasticity rule for this weight is proportional to the prediction error, which is the difference between the neural activity in layer 5 and the prediction that come down from layer 2-3 to layer 5. One of the studies that looked at how this weight synapses change from layer 2-3 to layer 5 is this study that they looked at what happened to this weight as they depolarize layer 5 neuron. And what they observed is that as they depolarize more, they get LTP in those weights, those synapses, and with a lower depolarization, they get LTD. And this is exactly what we would expect from our model. So we'll see as we increase the output of layer 5 neuron, we get LTP in those weights, and as we decrease the output of layer 5 neuron, we get LTD in those weights. But now a natural question is, okay, so let's assume that we have a prediction error in this network, how the error is going to manifest itself in layer 2, 3, and layer 5. One of the good study is this study by Jordan and Kerr that they basically put this animal in this virtual reality environment that the animal can freely run on this styrofoam ball. And basically the speed of a sliding of the grating on the wall is coupled with the speed in which the animal is running. And after training the animal, they start recording from parameter cells in layer 2, 3, and layer 5. And while the animal is running, they randomly halt the visual flow. And what they observed is that majority of the cells in layer 2, 3 showed depolarization effects, while this effect was completely opposite in layer 5, where majority of cells shows hyperpolarization. So we train our network on a synthetic visual motor task, where the input to the cortical circuit is visual flow, and we use the speed as a top-down contextual input to layer 2-3. After training, we randomly set the visual flow to 0, and we observed that the majority of the neuron in layer 2-3 shows positive prediction error signal, while the majority of cells or neuron in layer 5 show negative prediction error, which we interpret them as depolarization in layer 2-3 and hyperpolarization in layer 5. In that study, they also looked at what's the relationship between the mismatch responses and the speed in which the animal was running, and they observed that it is correlated. And this is pretty much what we see in our model, that the magnitude of the mismatch responses is highly correlated with the speed. To wrap up, I showed that self-supervised learning can be a common learning algorithm that can be implemented in neocortical microcircuit that allow the brain to learn different sensory modalities. I showed that it can learn a sequential task, and also discuss a couple of the functional benefits, such as denoising and being robust to occlusion, and also discuss two experimental data to support a synaptic plasticity rule, as well as the observation that we have from prediction error signal in layer 2-3 and layer 5. And I need to add that we have some predictions. It's at the end of the slide. Didn't have time to go over them, and we are totally open for adversarial collaboration. And with that, I'd like to thank everyone in the lab, and my supervisor Rui, Paul, and Loreen for all of the support. And the paper will be soon on bioRxiv. Thank you very much.
We have time for a few questions. We can start over here.
Really nice talk, and nice, I love the adversarial collaborations goal. Let's get them going. I'm curious about how you would think, because I guess the problem here is the time constant for the prediction is limited by the synapse delay through this one little thing. And I guess, as was mentioned earlier today, we see very different time constants in terms of general processing across the cortex, and we do predictions on very different time scales. So I'm curious how you guys are thinking about that.
Yes, so with regards to the time scale, what is really important is that as long as there is a bit of delay of information between layer 2-3 and layer 5, the prediction can happen in the future. So the only requirement is that layer 2-3 and layer 5 shouldn't be on the same time scale. With regards to how we think about as we go to higher cortical area, is that as we stack up this cortical column model, we're going to get one more delay. So naturally in our model, because of a delay from L4 to L2-3, it's going to add up. So if you have 10 cortical column, the 10th one going to predict 10 time steps in the future, but it might sound difficult because it's 10 time steps, but you're predicting the more higher feature as well. So if you see a cat now, probably in 10 time steps, you're going to still see cats. So, but this is something that we're exploring now. But in terms of time constant, I think one thing that's really important is there should be a little bit of delay, but the exact timing of it is not really that important.
We'll have one question in the back, then we go back to the front.
Okay, thank you for the talk. I have a question of anatomy. So you showed the input to the layer 5, but that's apparently in rodents and not in primates, because primates don't show this input to layer 5.
Yes, that's a good point. I mean, even if you look at the some experimental study, there are a little bit of differences between cortical areas as well. So, yes, I mean, it's a little bit tricky because the input can come from other part as well, and if it might be in mammal, it would be different mechanisms. But how layer 5 is going to receive the input, yes, in rodents it can be directly from thalamus, but in other brains it's not clear. But that's a good point. Thank you.
Hi, thanks. Hello. Okay. Thanks for the talk. Layer 5 pyramidal neurons have been in the spotlight recently because they express 5-HT2A receptors, which depolarize these layer 5 neurons in the context of psychedelic drugs. So I was wondering if your model might have any sort of predictions as to how classical psychedelics disrupt predictions.
Well, we have some predictions about optogenetic manipulation, that what happens in certain scenario if you do some manipulation in different cells of layer 5, the one that hyperpolarize, there are the few that depolarize. We haven't done that explicit prediction, but that'd be something cool to test as well. So we could definitely chat offline about it. Thank you very much.
Okay, let's thank all the speakers again. That concludes the sessions for today. Feel free to explore Lisbon.